So you've decided to have your DNA tested with an ancestral DNA testing company. You may simply be curious to see what parts of the world your ancestors came from, or you may want to get serious in researching your family tree and find as many relatives as you can. After anxiously waiting a few weeks, or maybe even months, you finally get an email saying the results are in. You do some initial browsing of your results, then decide to check out your matches. You see a list of names with various columns of information. How it's set out will depend on which ancestral company you tested with, but some of the information will probably just look like gobbledygook, particularly the one about shared DNA. For example, it might say shared DNA 348 cm. You might be thinking, does that mean if the DNA I share with that person is unwound and stretched out, it would be 348 centimetres, which is about 3.5 metres? Well, it might sound like there is some sort of logic to that. After all, DNA is supposed to be long if stretched out, but the full length of the DNA in one of your cells is only about 2 metres long. A bit bigger than that, but anyway. So that can't be the answer here. You may notice that the M is capitalised. With centimetres, the M is small. So M must stand for something else. However, the C is correct in that it does stand for centi. The capital M stands for Morgan, so CM here stands for CentiMorgan. What? You don't know what a CentiMorgan is? Well, I guess I better explain. As in centimetre, the centi part means 100th. So, just like there are 100 centimetres in a metre, there are 100 CentiMorgans in a Morgan. Now, to understand what a Morgan is, we'll need to have a bit of a history lesson. The Morgan, as a unit of measurement for DNA, is named after the American geneticist Thomas Hunt Morgan. In the early 20th century, he and his team established through experiments with fruit flies that the genes of inheritance lie on the chromosomes. Part of his experiments enabled him to not only map genes to particular chromosomes, but to their actual location on that chromosome. This involved taking advantage of a process of recombination between matching chromosomes. I'll try to explain this as simply and briefly as I can. Each cell of our body contains 23 pairs of chromosomes, so 2 copies of each chromosome, except for sperm and egg cells, which only contain a single copy of the 23 chromosomes. So during the process of cell division to form sperm and egg cells, the number of chromosomes is halved. So you might think that some chromosomes will be from the person's mother and the others will be from the father. But it's not as simple as that. During cell division, the chromosomes from the father will match up with the same one from the mother. They will join at a number of points on their matching chromosome. On average, this is 2 to 3 points on each chromosome in humans, but varies from chromosome to chromosome. The points at which they join up will also vary for each sperm and egg cell. The chromosomes then will swap parts of their chromosome, so some parts of the person's father's chromosome will end up on the mother's and vice versa. Through Morgan's experiments, he was able to calculate the probability that one of the crossover points would occur on a particular segment of DNA in the chromosome. The probability is not the same for all chromosomes or even different parts of the same chromosome. The point I'm getting to here is that the Morgan is actually a measurement of the probability of one of those crossover points occurring on a particular segment of DNA and is not an actual physical length. The unit of measurement used for reporting this probability is the CentiMorgan, with 1 CentiMorgan meaning there is a 1% probability of a crossover point occurring in that segment of DNA in each generation. The glossary in family tree DNA states that a CentiMorgan, CM, is a measurement of how likely a segment of DNA is to recombine from one generation to the next. A single CentiMorgan is considered equivalent to a 1% or 1/100th chance that a segment of DNA will cross over or recombine within one generation. For humans, 1 million base pairs, BP, average about 1 CentiMorgan. However, the rate of recombination is highly variable. But anyway, if a match has 348 centimorgans of DNA, what does this mean exactly? To explain this, I'll begin by referring to the explanations given by Ancestry DNA in their Ancestry DNA Matching White Paper. In this, they give a basic 5-step process they use when comparing two individuals. So I'll just read, or I'll paraphrase what they say. Okay, so they break each chromosome up into short segments, which they call windows. Each of those windows contains exactly 96 SNPs, which is often called SNPs. Each SNP, single nucleotide polymorphism, is in effect a single base pair in the DNA. In other words, one of the letters in the 4-letter alphabet in the genetic code. Now, then they, for the second step, they take two individuals, or for each pair, they will then identify the windows, so those short 96 segments that match. And for those, they call them a seed, so they've matched the two individuals. Then for the third step, for each of those seeds, they will attempt to extend the segment So they'll carry on where they've matched those segments. They'll carry on for the two matches and identify where they're identical, and they will continue to do that until they either reach the end of the chromosome or they no longer match. And they will then call that a segment that is an IBD. In other words, they're identical by descent. So it's thought those segments come from a common ancestor. In the fourth step, they calculate the length of that IBD segment in CentiMorgans. So in other words, they're calculating the probability that the IBD segment would be broken in a single generation. I'll just mention here that the longer the segment is, suggests that it is from a more recent common ancestor, because over the generations they would tend to be broken up into smaller and smaller segments. Okay, we go on to the fifth step, and if the segment is longer than 6 centimorgans, so in other words there's a probability of more than 6% that in a single generation it would be broken, then that segment is considered a match and is stored in the database as such. Now, if I go on a bit further, it will explain why they chose 6 centimorgans. Okay, I might just read what they say here. The cutoff of 6 centimorgans was chosen after considering several factors. The first factor is data storage. Since the number of matching segments grows exponentially with decreasing length, we dramatically reduce the storage requirements of our matching database by increasing the cutoff point. So in other words, it's basically their ability to store the data that they cut off those little bits. Otherwise, it becomes very voluminous and slows the system down. Then the second part, and this is the more important, is that the accuracy of the IBD segments drops off more significantly as the length decreases. That is, the shorter the length of the detected IBD segment, the less likely it is that the detected chromosome segment is truly inherited from a common ancestor. In other words, those smaller segments could simply just be a coincidence, or as a result of a mutation that has occurred in that person. So anyway, after all this is done, the length of the segments are added together to give the total length in centimorgans. So these are the segments over the 6 centimorgans that I spoke about. The other companies use similar methodologies. Additionally, most of the companies will also list how many segments there were that give the total shared DNA and the length of the longest of those segments. Long segments increase the probability of a recent common ancestor. Now, personally, I have my parents and my DNA listed with 4 ancestral companies. And from looking at the total matching centimorgans I have, with my parents, it appears the possible CentiMorgans ranges from about 6,800 to 7,200 CentiMorgans, depending on the particular company. The actual full length is thought to be about 7,400 CentiMorgans. These differences could be for a number of reasons, such as the referencing of different research for calculating the probabilities for the crossovers, which then results in slightly different lengths of CentiMorgans. There could be variations in the sequences of DNA tested between the different companies, as well as differences in their methodologies for calculating the total CentiMorgans. I should also note here that there is a genetic factor in the frequency of crossovers, and it does vary from person to person. It has also been shown that the frequency of crossovers is lower for the formation of sperm and higher in the formation of eggs. So the total centimorgans in males is lower than the average, but is higher than the average in females. I believe the companies use an average in part because it would be much more complex to factor in the differences between males and females. But the main reason would be they do not know exactly how you are related or how many men and women are between you and your match's common ancestor. Of the 4 companies I have shared my DNA with, Family Tree DNA and MyHeritage also have a chromosome browser where they map all the shared segments to their positions on the chromosome. It appears Living DNA are planning something similar in the near future. I'll leave it there for now, but in an upcoming video I will discuss how the amount of shared DNA can be used to assist in determining how you and your match are related. So make sure you subscribe and click on the bell to be notified when I upload future videos. If you like what you have seen, click on the like button and keep watching for new videos. And if you really love my videos, then click on the like button and help support me to make more at Patreon. I'm David A. Elliott. Have a good one and see you around like a Rissole.